Good morning. My name is Vern Smith, and I'm working with my son Ford, and we started a company called Regenerative Land Solutions. It's accumulation and a process of a paradigm shift we had where we're trying to look at growing crops through the biology first versus the chemistry first lens. It's kind of a journey we've been on. We want to share with you guys some of the results we're seeing and see if it's possible we could help you guys as well on this journey. What we've come to realize is there's a whole world underneath our feet that sometimes gets overlooked. And all these little guys will work for free for us if we take care of them. And they're very significant in helping our bottom line. And we're trying to educate people on this and also educate ourselves. It's like a new frontier, but it's pretty exciting. And we're thinking we are seeing some progress in this area. The gal that first pioneered these thoughts and brought it to the forefront in people's minds is a lady named Dr. Elaine Ingham, and she made this chart here. And this is the different trophic levels of the plant. So we start out with photosynthesis, and the area that we're going to address right now is the second trophic level, which is, and by trophic, I mean how a plant eats. So we're going to focus, if you can see the cursor here, on the fungi, the bacteria, and the protozoa. Simply in nature, most of the nutrients that go to a plant come via the source of the fungi and bacteria, and they act as miners. They go out, they take the different things that the plant needs out of the soil. The plant says, well, I need some molybdenum, and then so it'll secrete sugars which will attract whichever fungi or bacteria has consumed those and got them in a system. They'll come colonize in the rhizosphere. Then the protozoa, which are your amoebas and flagellates and ciliates, when they consume that particular fungi or bacteria and secrete it out of their system, that's perfect plant available nutrients. So what we're trying to do is see if we can keep this system active, keep the synergy going between the microbial community and the plant community above ground. When we look at what is soil, it's interesting that half of what soil should be is air and water. A lot of times we miss that. The organisms that do a lot of the work, they are a very, very small percentage, but without that air and water, they can't thrive. And when they thrive, they also increase the organic matter, which everyone is trying to do. And we'll see that in a slide a little bit later with a study with MSU. I really like this slide because it shows how plant succession is a function of the fungal bacteria ratio. In nature, when you have a catastrophe like a flood or a volcano or a mudslide, and it wipes everything out, the first thing that gets established back in that system is the bacteria. So we would call that ratio very bacterially dominated. It'll be well below 1-to-1. The plants come in, those early successional plants in an agricultural situation are plants that prefer their nitrogen in the form of nitrite, and we call them weeds. We don't really want them. When you look here in that yellow box, that's the target range we want to be for most of our grasses and row crops. We don't do a lot of citrus and almonds in Montana, which they would want a system that has way more fungi than bacteria, but we're shooting for 1-to-1 ratio. So the fungi, in order to get that fungal count up, to get that ratio where we want it, we have to be mindful of what damages the fungi and how we can mitigate it and get it back into the system again. And the things that do damage the fungi, since it's more sensitive than the bacteria, is disturbance, excessive fertilization, excessive herbicide use, overgrazing is a big deal too. So all those things hurt us, and if we can learn to mitigate against those, we'll see our systems become more efficient. When we look at the effects of good soil, this is just a few things here, but the ripple effects just seem to go on and on. If you have good soil that's getting all the proper macro and micronutrients, you're going to have a more nutrient dense crop, which will in turn, it'll put weight on your cattle. It'll make people healthier if they're eating it. Also, the water holding capacity slide shows that in enhancing your soil biology will increase your carbon. Well, every molecule of carbon will attract 16 molecules of water. We want every molecule of carbon. Also, when the soil's healthy, there's more space for that water to be in there and more air too. And that's a function of the fungi because it'll build your macro aggregates. Fertility and germination rates will increase. We have a slide coming up from a pivot we worked on in Dry Creek that demonstrate this quite nicely. Disease, insect pressure. I mean, last year we had a terrible year for grasshoppers. The grasses that had the higher bricks seemed to suffer a lot less than the plants that were suffering anyway. And the insects will go to diseased plants a lot quicker than they'll go to healthy plants. And a lot of systems we're working with in different parts of the country, disease pressure is mitigated immensely by working on their biology. So if you can have a system that's healthy, you can diminish your inputs. That improves your bottom line. We talked about healthier food for people. Clean water, the soil cleans the water. If the water is able to infiltrate through, the water that gets into our rivers, into our aquifers will be a lot cleaner as well. And the weed pressure, that's a big one. We got a few slides coming up that will demonstrate that, but the balanced soils will not encourage the germination of these weeds. Fungi is the toughest player to keep in the system, but it also plays one of the most significant roles. One of the driving forces is the fungal bacteria ratio, like we looked at on the chart above, but also how much activity that fungi has. Does it have the proper food it needs to function? Does it have the proper air and water? If we can get those two going for us, a lot of other things seem to fall into place. And how do we measure that? Well, here's an example of a test we did on a field in the Paradise Valley. The guy's issue was that his fields just weren't producing. He was running about 50%. This test is a test that we use to determine what the biology is. It's using a testing system that does direct microscopy where they count every single thing under a microscope. It takes a highly skilled technician to do this, but it's pretty compelling what we see here. So I'll put my cursor up here. The total fungi numbers are here and the bacteria is here, which gives us a pretty decent balanced system. But when we look down here at the activity levels, which I talked about on the previous slide, we're deficient there. Well, the fact that the fungi is not working makes the system almost behave more like a bacterially dominated system. And without the fungi building structure for the protozoa, we see those numbers are deficient. And when we take all these things and put them together, we can figure out what the nitrogen cycling potential is. And we have here it's 100 to 150 pounds per acre. On a normal system that's really functioning well, that number should be 300. He worked with us, saw some significant results, and we're kind of excited to see that what we saw underground mirrored what was going on top, and we were able to solve the problem for him. We worked with MSU and with the Arthur Blank Foundation at Mountain Sky. They were very generously helped support us on some trials we did there, and I'm going to run through some of the 2020 updates we had and things we saw this past summer. We set these trials up in 2018 and replicated for them so that potentially we could have this written up in a peer-reviewed paper. It's a little more scientific if it's not a side-by-side. Side-by-sides are huge too, but this is for academia. They prefer this system. We had two treatments that basically we ran throughout the course of the trials. We had compost in 2018, but we nixed that because it just was not feasible for a producer to use in this country. So the two products we use, the one that we work with is Provide and Revive. It's in both of these products, Spectrum and Nutrinate, there are compost teas that have a food supplement that they use to enhance them. The difference between the product we use is it has a lot of diversity. It comes out of the wild. It's got a stable shelf life up for 2 years, but it doesn't, it's not raised in the lab. Like the Spectrum is raised in the lab and they have probably a dozen or so different species in there that they think will help. We prefer, from what we've seen now, to try to get the most diversity we possibly can underground. They've been doing a lot of studies, and it indicates that there's a lot of keystone players in systems that if they're missing, even though they may be small in numbers, their significance, their lack of presence can have significant results. We'll go with you a couple graphs. Organic matter, that's carbon. We talked about water holding capacity. The higher the carbon, the better chance you have of hanging on to the water that gets into the system, whether it's rain or irrigation. The treated greatly increase that compared to the control. That's significant. One of the driving forces in creating organic matter is the biology in the soil. Also, the way a lot of issues we have with phosphorus is it gets tied up. That's frustrating. You spend good money on it, you put it down, it gets tied up and the plant can't get it. What we see in this graph here is that enhancing the biology frees up the phosphorus so the plants can get it. That's just money in the bank. Everyone wants more feed and they want it to be more nutritious. That's just common sense. These graphs demonstrate it quite nicely. The product we work with, this is a significant increase in dry matter. This is also a significant increase in relative feed value compared to the control. The last item on the benefits of good soil is the weeds, less weed pressure. This is an interesting study we did up Dry Creek on the church pivot on Mountain Sky, where they allowed us on a new seeding to use half of the pivot the way we wanted to, and the other half of the pivot they just did status quo with Rocky Mountain. We're not against fertility, but we're very much trying to get people to be judicious in the use of it and try to get to some less salty fertilizer that's more biologically friendly. So we went off of the chemistry and the biological testing, and we came up with a package where we used the Earthforce Provide and Revive, fish hydrolysate, and some G22, which is a nature's product that Mark Narden in the valley here sells. And our package came up $2 an acre cheaper up front than the conventional. And we predict that that'll cheapen up over time. But here, I'll show you some of the results we saw from that. We laid out a grid pattern with some hoops to count the weeds after the field was up. The picture on the lower left obviously looks cleaner than the one on the right, but looks can be deceiving. So we got on our hands and knees, went through the hoop, hand picked and counted every single weed in every hoop and extrapolated it over the course of the field so it could be per acre. And when you look right here, there's a significant difference in the weeds in the treated compared to the traditional. And if you want to take the time to look at the different species, we also count it and measure that as well. Well, if weeds are going to germinate and die off, there's going to be less of the plants that you want to be in there. I also talked about for germination rates, trying to get the biology right. This particular field, before it was seeded, was fairly bacterially dominated from historical uses. If you look at the control on the left, there was significant more bare ground on the left than there was on the right of a treated area. That grass is cash meadow brome. That's the plant they wanted. And the alfalfa, we had a higher germination rate because we mitigated the problem with the biology up front instead of afterwards. Long term, this is way more production for the farmer, because once you get something established, till you reseed and farm it up again, you're pretty much going to just have to live with what grew that first season. Another project that we didn't start on, but we came in afterwards, was in Belgrade. This gal bought some ag land. She wanted to seed it back. So there was a irrigation ditch which ran between the two fields. She seeded them late, had a fairly successful germination rate on the top left-hand corner. And this was an alfalfa grass mix, but in the lower right-hand corner, it may look like it's a more robust field, but it was a solid stand of kochia. Rather disappointing for her. But what's interesting is when we went back hindsight and did some tests there, the lower right-hand corner with kochia had a fungal bacteria ratio of 0.39, and the one up on top had one of 1.27. What we see here is what was underground was mirrored in the successes above and the failures above. So if that had been mitigated ahead of time, she would have had better success. This shows the same fields in the fall. She wasn't too discouraged with the top field. It wasn't irrigated. It was a dry land deal. It'll work. The bottom field, they're going to have to start over. And she inoculated, worked with us, and we'll go back and photograph that field probably next spring. In the West, a huge issue is the non-native annual bromus grasses. We call them cheatgrass. In Texas, they have a similar species called rescue grass. This paddock was solid rescue grass and a few broadleaf weeds. We inoculated the one side and not the other, and seeded everything at the same time. So when you look at the treated side, the rescue grass struggled to germinate. It's hard to see in that picture, but there's a lot of the perennials that got established. On the left side, where we didn't treat it, the rescue grass was very healthy, didn't have any problems at all. And that's not what this lady wanted, and that's not what we want. We're also doing quite a few trials on cheatgrass, and we're seeing that some significant results there. If I had to pick soil, I couldn't have picked a worse place to do a trial than this. This is down in my neighborhood. There was a bridge project, and the field that you're looking at is basically the substrate from the county road that the bridge builder bought in. They filled a hole, and I thought, you know what, I'm going to try to grow a perennial pasture on a parking lot. If you look at this picture here, I took a 12 seed cover crop mix. I mixed in some perennial grasses that I wanted. I had a hydro seed company come, and we were going to use a thin layer of hydro seed to glue the seed to the ground. I didn't even try to work it in. I didn't try to graze on it. What I wanted to do is mix in the biological inoculants in with the hydro seed, and I didn't realize the guy was going to have to fill his truck 4 times. So I put all the product for the whole project in the first tank. And so when you see this picture, the right-hand side had 4 times the recommended dose. The side on the left was just 1 gallon, 1 pound per acre. The left side did establish, but it didn't establish near as well as the right side. I don't think anybody's working with ground quite this bad or quite this sterile, but it's cool to see that it worked. We have in my fields out in front, we did a side-by-side comparison. I did some treatments. I did 2 of them before the fall of 2018. It's area that I've seeded. I seeded, I watered it, and I grazed it the same way for 20 years. Jed Eberly came down, took some soil samples back in 2018, and he was able to run the DNA sequencing on the bacteria this summer, and he was pretty excited to see the results. So the issues I struggled with, I had an acidity problem. The fields were fairly productive, but I was struggling to keep them wet going through. It's very sandy, very dry. Some of those things I'll talk about in a minute, but what was cool was when he did the DNA sequencing, the diversity was way higher in the treated field compared to the control. And also that the biology that was in the control, the taxa, was associated with unhealthy acidic soils, where the side that was treated, the taxa in there was indicative of healthy soils. What was interesting is, even though I tried to graze it correctly for 20 years, I didn't do anything to change the diversity underground. I think if we think that our practices are going to change that diversity in a short amount of time, I don't think that's happening. I think this takes decades, possibly even centuries, to change, because where are the new species going to come that we've damaged? You know, if we can inoculate them like we do a probiotic and a prebiotic after penicillin, we can greatly speed this process up. And this was a good example of it. Living Sky Greens, which used to be the old Wheat Montana, Frank worked with us. He did a bunch of trials. He did 14 trials in 2 of those plots. He worked with us with our Provide and Revive. It was a particularly bad year. They didn't get much rain. Our 2 plots got put in after a snow event, and then no more moisture came after that, which is just the luck of the draw. No big deal, but we actually saw better results than some of his other trials. He'll be working with us again this summer. Those yields aren't good, but they didn't get any rain either. So what the effort he put into there, that residual will carry on through this year as well. California, it's going to be hard to read this, but the takeaway, if you can see this, is that one of the neat things we see about improving biology is the way that the water is used by the plants more efficiently and less water is needed. In California, when you go to harvest almonds, you need to stress the trees by pulling the water off. Typically, it takes a week, and then when the tree starts to stress, and they use some very sophisticated equipment to determine when, they go, okay, now's the time, and they'll shake the almonds off. Well, the plots we worked with, instead of the usual 7 days, it was 21 days before they started showing stress. That's pretty neat. Last slide I'll show just happens to be a neighbor, really likes her landscaping. I said, you know what, I'll set you up with a backpack. I'm sure she sprayed them heavy. I said, you have to save one plant and don't spray it. So on the lower right-hand corner, there's one plant, and then the picture on the top left, that is just 2 plants. And this is after, about a month or so after being sprayed. We're excited about this journey we're on. We have a website. We're constantly putting more things on the blog, more case studies. If you want to get a hold of us and you're interested in working with us, there's our website. Appreciate your time to listen for this. Thank you.